incoming assistant professor with South Dakota State University, but I'm currently a T32 postdoctoral fellow at Penn State. I am housed within both biobehavioral health as well as the Center for Healthy Aging. Before that, I had my master's and PhD in human development and family studies in Oregon at Oregon State University. And for my bachelor's, I got actually a bachelor's in psychology in a small university in southern Utah. So a lot of my work really kind of runs the gamut across psychology, HDFS, as well as behavioral health. Okay. I'd also like to take a moment to acknowledge the land that we stand on. So normally I would, usually being in person, I would acknowledge the land that everybody is standing on at the moment, but because we're all at different institutions right now, we're all kind of zooming in with the approval from my co-presenter here. I've just kind of copy pasted both Penn State and UCSF's, which is where we are currently, their land acknowledgements. And I'd like to just take a moment and honor the land that we are currently standing on, that we do work on and research with. Importantly, a lot of our institutions partner with Indigenous communities to write these acknowledgements. So if you haven't, I would encourage you to explore your own institution's websites to better understand the land and where it's come from. Okay, so jumping in to research. I've really been fascinated by this idea for years now that although daily stress comes in many forms, it's really this day-to-day living that wears you down. In fact, we in our lives often move from day to day. We live our lives not thinking about the future very often. So something that's really ingrained in my career is that the idea that if we live our life from day to day, why aren't we studying day to day? So this idea of examining daily stress or the daily side of stress has been around for a while. One conceptual model proposed in 2005 by Dr. David Almeida here at Penn State and revised this year by colleagues is the daily stress process model. Now, the daily stress process model essentially suggests that characteristics and appraisals of daily stressors inform daily and long-term well-being through stressor responses. So for the purposes of this presentation, I'm focusing on these affective responses to daily stressors, but it's important to note that there are other types of responses that we have. You have sleep, we have our biological responses, that sort of thing. Affective responses, however, refer to two temporally different phenomena. Seen here in kind of this blue box, these two conceptual phenomena are reactivity and residue. First, affective reactivity refers to these same-day changes in affect. So, for example, you have an argument with your spouse, you become angry or upset in some way. This is not limited to negative affect. Indeed, positive affective reactivity is a similar process, but instead of this increase in stressor, in negative emotions, you see this decrease in positive emotions. Second, our affective residue refers to these kind of next day changes in affect. This is often operationalized as the association between your affect in relation to a previous day stressor. So in the example that I provided, you had an argument with your spouse and you go to bed angry. You wake up the next day and you're still upset about whatever that argument was about. So to put another way, affective reactivity refers to a more proximal initial change in affect, and residue refers to a more lingering or continued change in affect. Unfortunately, although theoretical perspectives have highlighted both of these things as important, the majority of research has examined how affective reactivity is associated with health outcomes. So literature has suggested that these same day increases in negative affect that constitute affective reactivity are related to things like biological health, like elevated inflammation, cognitive health, like higher response time and consistency, mental health, like increased depression risk or depressive symptoms, and behavioral outcomes like marital risk. Conversely, to my knowledge, at least, only a handful of studies have clearly articulated or examined how this affective residue relates to health outcomes. So these two studies were kind of the only two that are out there to my knowledge. One's by Kate Ledger and another's by colleagues of mine as well as myself. And we found that these next day changes of negative affect, these lingering feelings of negative affect, have implications for both functional limitations as well as increased depression risk. So this evidence becomes important when considering targeted intervention space. Not only are affective reactivity and residue daily indicators of well-being, but they have been acknowledged as targets for possible behavior change. So seen as in this conceptual model specifically, affective residue is this component that plays a role in the longevity of a stressor response and the potential impact of pileup. And this has implications for things like allostatic load and other implications that are better articulated in the paper. So I won't bore you about that. But given the lack of empirical evidence for how affective residue informs health that I just presented, one of the things that I began to wonder in my career is, could these researchers just simply be wrong? Like, this recovery or residue period, does it just not exist? That there's nothing there and that's why no one's publishing on that? Or is it just elusive? And if it's elusive, why would it be that way? So I began to look into this idea of affective residue a lot more thoroughly. So to preface, all of the results and examples I'm about to show are from the Midlife in the United States study, or dataset, or otherwise known as MIDAS. MIDAS is a national and publicly available dataset with 3 waves of data spanning every 10 years, and it began in 1995. In 2011, the MIDAS team also recruited a refresher sample, that's what they call it, that would allow them to compare cohorts forward. In particular, these results that I'll present are all from the National Study of Daily Experiences, which is the 8-day daily diary subset of participants who completed that. The sample includes a little over 2,000 participants, and they completed surveys at the end of the day for 8 days. These surveys were actually telephone interviews about their daily experiences, included but not limited to daily processes, health, and positive activities. And so I've included the website down here at the bottom for more information if anybody's interested. So affective reactivity became this puzzle, and I became focused on trying to understand why and under what conditions do affective residue occur. As such, I turned to the daily stress process model again to try to make sense of this question. Now, there's some work out there on daily stressor characteristics surrounding daily stress. Aspects like type of stressor, who is involved, these aspects may be important considerations for affective reactivity and residue. But the thing about those characteristics is that they're not really modifiable. So simply put, by definition, daily stressors are these short-lived, commonly occurring negative experiences that people encounter just living their lives. Some of these, like arguments, you may be able to avoid with like a spouse, but may not be able to avoid with a coworker or a boss, for example. But other things like getting stuck in traffic or having a work deadline or presentation to strangers or having a dog bark while presenting things, they're unavoidable in daily life. Appraisals, however, about these daily stressors are modifiable. So one such appraisal is resolution. Resolution refers to this subjective indication that a daily stressor is no longer ongoing and is often measured through a one-item question of, is this issue resolved, when asking about particular daily stressors. Importantly, resolution may be related to the downregulation of emotions. and as such may be related to diminished or extinguished residue. So when you combine these things, ignoring resolution, you may be not capturing residue in its entirety. So one thing led to another, and in a 2021 paper, myself and Dr. Robert Stosky, my mentor, tested this idea that resolution may be related to diminished or extinguished residue. Now, to orient you, this graph here shows the estimates for arguments specifically related to changes in negative affect. The dark gray bars are when an argument is unresolved, while the lighter gray bars are when an argument is resolved. The differences between the bars within each of these red boxes were significant. So that suggests that resolution did significantly dampen both this affective reactivity and affective residue components with arguments. What I believe to be particularly important here is that although resolved argument bars are still significant, suggesting that they dampen reactivity but don't eliminate it, the resolved bars for residue, however, were not significant. So although resolution significantly decreases this reactivity, it may eliminate affective residue related to arguments for negative affect. Therefore, as I said, when examining exposure to arguments, as many do, we often ignore things like resolution or other appraisals. And so we may miss the nuance that's associated with residue. Now, another modifiable appraisal is our stressor control. This refers to the average perceived control over daily stressors, although a small number, previous literature has explored how daily stressor control varies by the type of stressor. So for example, control over an argument might have different implications than feeling control over a work deadline. Most work, however, has focused on characterizing how perceived control of these daily stressors simply changes over time. Indeed, work from a colleague, Dr. Eric Serino, has found that perceived control decreases over 10 years, and that these changes do indeed vary by type of stressor. Importantly, stressor control might be a resource. If a person feels more control over a stressor, they might be able to better cope with or may be less likely to ruminate on these events, and therefore have dampened emotional reactions to them. The following question then that my colleagues and I had was, if perceived control might vary by stressor type, does control have differential impacts on affect depending on that type of stressor? Now, it's important to note that because all stressor domains were included in this model specifically, the intercept then represents no reported stressor occurring. And it's kind of difficult to see given how many bars are here, but perceived control moderated some, but not all stressor types and negative affect. So you can see on the left that there was a general pattern across stressor domains. So starting with arguments, we would interpret these by saying on days when an argument occurred, having more perceived control over this argument was related to less affective reactivity or alternatively smaller same-day increases in negative affect. Similarly, on days when avoided arguments occurred, there was smaller increases in negative affect as well as with work stressors, with home stressors as well. There was only one stressor that really showed any signal for this affective residue component. Specifically on days when a work stressor occurred in the previous day, there was a significantly smaller increase in negative affect when perceived control was higher compared to lower. So perceived control did seem to attenuate the effect of work stressors specifically for this negative affective residue. So these examples are particularly important, bringing it back to potential interventions and behavior change. So importantly, if we can determine when and under what modifiable conditions reactivity persists into residue, we can determine what aspects of daily stressors are pertinent for intervention. Now, daily stressor resolution and daily stressor control are just two examples that I've been working on, but there are others, things like perceived severity, resource risk, that may be additional modifiable aspects of daily stress that inform stress responses more broadly. Moreover, what these results might underscore is that we as researchers should be moving beyond simply exposure to daily stressors or simply just your same-day affective responses. There's strong evidence that daily stressor exposure has implications for health and well-being, as well as affective reactivity. But I believe that daily stressors are, as the villain Thanos would say, daily stressors are inevitable. Understanding the aspects of daily stress processes that can be modified might lead to new discoveries across research and intervention spaces. All of that said, much more work is needed to characterize the impact of affective residue and possible mechanisms through which residue occurs. Theoretical perspectives underscore the possible implications for health and well-being. However, not only 2 studies to date have clearly articulated how residue is related to health. Future work will need to test how affective residue is associated with health and well-being outcomes outside of just functional limitations and depression. So in line with that direction, I recently submitted a grant to the National Institute on Aging to test how affective residue was related to inflammation. Further, we need to better understand what conditions and for whom both resolution and control may be a buffer for daily stress and residue processes. So one future direction might be to examine resolution and control in conjunction to better determine their unique combined associations. There's also the component of individual differences. So some of the work that I'm currently doing, and maybe I can work with Ryan, shout out to Ryan, who's coming next, can work on with is looking at how affective responses within marriages are modified by marital satisfaction or marital quality. That's just one example, but there's tons more to do on this research. With that, I'll throw out a few acknowledgements. Everybody who's worked on these projects with me, Dr. Rob Stosky, Kelly Chandler, Eric Serino, Jennifer Graham England, Dr. David Almeida, as well as funding, all of the funding that comes from these projects, and of course the universities that I've done this work at. And thank you.
Thanks, Dakota. That was super interesting, and fingers crossed for your grant. I hope you get it.
Thank you.
Our next speaker is going to be Dr. Ryan Brown. She's currently a postdoctoral fellow at the Bakar Aging Research Institute at the University of California, San Francisco, and is co-executive director of the NIA Stress Measurement Network. She received her PhD from Rice University. And Dr. Brown's overarching goal is to investigate how fundamental human experiences like love and loss contribute to mental and physical health as we age. This includes adopting a lifespan orientation, psychoneuroimmunological perspectives on social loss. Dr. Brown's also active in social communication spaces, and her talk today is entitled Stress, Aging, and Relationships, Conceptual Considerations from an Integrative Perspective. Please join me in welcoming Dr. Brown.
Awesome. Thank you for that warm introduction. I'm really excited to chat with y'all today. I think this is, as we were alluding to, is an exciting time as we're entering our first faculty jobs to be thinking about these developing research programs and also potential collaboration. So a lot of what I'll be discussing today is how I think about love, loss, and aging, and using stress as a lens through which to sort of connect those pieces. And I really like to think about these malleable biopsychosocial factors that we might be able to leverage. And I've really appreciated a lot of Dakota's work on the daily stress processes. So you'll see that I think the way that we reference each other here really speaks to a consensus sort of in the field of this need to go really zoom in on a lot of these processes on a more daily level. So just briefly, as was alluded to earlier, I do absolutely think across all of my studies with this lifespan perspective, my graduate training was focused on the death of a spouse and considering bereavement-related processes. especially stress response processes through this time, and really thinking about using attachment theory to better characterize what we might expect for stress response processes after the loss of a spouse, and considering early life experiences as well. So although this is not the focus of my talk, I just wanted to emphasize that you'll probably get that lifespan feel throughout a lot of the topics today. So I'll start off by just highlighting some resources that we have available through the Stress Measurement Network that I think can be really helpful for you active researchers to share with students, et cetera, and then discuss some considerations I have around stress and aging research, as well as stress and relationship research, and then talk through a couple of conclusions and points for collaborations. So first, in thinking about resources that I really thought we should highlight and the ways that we can advance our science of stress processes, a really critical way that we need to do that through our journals is having a shared language and talking about stressor exposures, responses, the timescales, the life periods, and any particular characteristics of those responses that are really critical. So the Stress Measurement Network has this stress typology guide on our website. It's stressmeasurement.org. So I just wanted to direct folks there. I've heard that this can be a really useful starting point, especially if we're working on projects with students, or if you're getting into a different area than maybe you were trained in. The other resource that is really helpful, especially for newer measures that we might be focusing on, like climate stress, is these entries on measurement and recommendations, whether that's psychological measures like something like climate stress, which we have added recently, as well as physiological measures. We also offer consultations. So if you're working on a grant and want feedback on either measures or study development, you can always reach out to us through the website. And then what I'm personally most excited about is doing more with science communication and dissemination. So we'll be launching a podcast called The Stress Puzzle in this upcoming fall. And I really wanted to highlight this because in my mind, the most frustrating or place coming up as a grad student that I felt like, oh, I just have to play catch up so intensely, is just understanding the historical context of stress research. So I was really hoping with this podcast that we'll be able to pass more of the context of where we're at now as a field to other folks coming up, and really promote the highest quality stress research that we can possibly be doing without sort of reinventing the wheel and going back to the drawing board, and bringing up ideas that have already been sort of discussed and debunked in some way. And then also fostering openness and transparency around these processes. And, you know, in this time where we see a little bit more anti-science aggression, trying to emphasize like the how and the why of these studies. So I wanted to emphasize this because we're at a really critical point of development right now, where any folks here who have feedback on what kinds of topics, resources, historical context do you see that your students might struggle with, or that you yourself coming in from a different angle would appreciate hearing and getting that perspective, I would really appreciate any feedback there. So I'll have my email on the last slide. Feel free to reach out. And then going into more of the research specific side, I really wanted to emphasize why I think now is a really awesome time for folks who are interested in thinking about health and aging processes to be stepping in from the social personality and health space. And that's because aging research has focused so specifically on biology for so long, right? And that's understandable. We see these biological hallmarks of aging that are really related to these age-related health conditions. So we might be thinking about cellular senescence or telomere attrition. And, you know, there's really cool work where you use model organisms, make a genetic mutation, and you can, double the lifespan of a worm. But as we see when we translate this kind of work to humans, it gets a lot more complicated. So I've been really compelled by some of the work done by Eileen Cremens, highlighting that biological variables certainly explain some of the variability in aging outcomes, but highlighting also the benefit that we get when we also add in the social variables. So just briefly, I wanted to show you, this is from a sample of over 1,000 nationally representative older adults. I believe it's the Health and Retirement Study here in the US. We're going to, on the next slide, to see the variance explained for multimorbidity, disability, cognitive dysfunction, and mortality. I'll probably focus mostly on multimorbidity and mortality. And I really want to highlight right off the bat that we're not even getting to 25% of the variance explained here in mortality. And that's where we have the most variance explained. So when we just look at the biological and the demographic components of aging, we're not even getting a quarter of the variability in mortality. It's less for cognitive dysfunctioning, multimorbidity, and disability as well. And so here you see there, some of these are really more biologically determined, whereas something like mortality, your chronological age matters a lot, shockingly. But what's exciting to me is when we add the social variables in. So let me make sure my slide advances. So when we add the social variables in, we get further along here, right? We're a little bit over 30% of variance explained. And what's really striking, though, is that when we look underneath this, we can see that the social variables explain more variance across all of these outcomes. So the social variables are the ones highlighted here compared to the biological variables. So for disability, we see 6 times the variance explained, 36% more variance for multimorbidity, almost 5 times for cognitive dysfunctioning, and 1.8 times for mortality. So we're really not fully explaining how these social variables are affecting health through biology, right? Because if we had this perfect one-to-one line of this social things happens to this biological process and then aging or mortality happens, right? We don't have that clear process. And many age-related studies are omitting social variables or not focusing on them. So this has gotten me excited that there is more of this call to integrate social components into thinking about the aging process beyond just the biology. And based on the kinds of questions that I'm interested in, I've been really interested in in my postdoctoral training and getting a better understanding of the relationship between aging biomarkers and aging processes more generally. So there are a couple of options of how aging biomarkers, like something like telomeres, and I'll bring up another longevity factor called clotho in a moment, but how are these different aging biomarkers related to each other? Is it just because you're aging and as you age, everything is being downregulated? Are one regulating one another, or are they being regulated through common processes, like something like chronic stress? So I was interested in looking at this in a sample of caregivers that was collected here at UCSF. And so the research question here was if chronic social stress might promote this coordinated downregulation of longevity factors. And importantly, all of the participants in this study were caregivers. The distinction was the level of neurodevelopmental need that they were attending to with their child and the level of perceived stress that they had. So we stratified our sample into high stress and low stress based on those characteristics to examine how something like telomere length might be related to something like clotho. And clotho is this aging biomarker that really acts as a circulating hormone. It extends lifespan in model organisms. So it's one of those that you can go in, do a genetic mutation with a mouse and extend lifespan. You can mess with it and really shorten lifespan as well. So we don't need to think too specifically about these 2 markers, but I want us to back up and think conceptually about what this might mean. And what we really found was that we see this evidence for a downregulation of these longevity pathways through chronic social stress, but only for the folks in this high stress group. So the low stress folks, on the x-axis here, you're seeing clotho levels, and on the y-axis, you're seeing telomere length. And so for the folks in this low stress group, there's no relationship between these 2 aging biomarkers. But for the folks in the high stress group, we see in PBMCs as well as in more specific cell types. So we can do these really fine-grained analyses to look at the specific types of T cells and B cells. We can see that these aging biomarkers are only associated with each other for folks in high chronic stress conditions. And that gets me thinking both about this moment to be thinking about aging research as you know, someone really more socially driven, as well as the gap in our understanding of relationships contributing to aging. So another study that caught my eye more recently has been looking at social relationships as predictors of accelerated aging. And again, another nationally representative study with a large sample of older adults, over 3,000, and they found that accelerated epigenetic age was associated with folks who did not have strong social relationships or had lower social support within those relationships. And to me, this points to, we see this impact of relationships on, we see this association of relationships with aging, but we really are so zoomed out that understanding the processes through which that is happening, it isn't possible from these studies. So as we switch sort of into this stress and relationships section, I want to think about how the death of a romantic partner might influence aging trajectories. And the first study I'm going to show you will not answer this question directly, but I wanted to give you this question of broader context for how I'm building and thinking about these research questions. So thinking about the death of a spouse or general social loss experiences like divorce as well, as potential moments where health behaviors may change. We lose the impact of our relationship partner on distress management and many other components. So I'm going to come back to this idea of this event and the time after it and what that adjustment process might really mean. But first, let's just look at and this in the context of an acute social stressor and thinking in terms of the level of grief severity. So we've seen from prior research that things like loneliness and depressive symptoms can sensitize your inflammatory response such that when you face a stressor in the future, you have a more pronounced inflammatory response to that stressor. And we think that that might lead to greater cardiovascular risk over time. The death of a spouse is thought to promote this biobehavioral dysregulation that can amplify stress response processes. And so I was looking at persistent high grief as one index of this dysregulation. So here I wanted to look at if high grief symptom severity might sensitize one's inflammatory response to future acute social stress. And this was as part of my graduate advisor's R01 looking at the first year of bereavement. And I want to emphasize that because I think it's really important that this, again, such fundamental life experience of the death of a romantic partner in older adulthood, right? We're not even thinking about an atypical timing. There's so little work in this first year where we see the greatest health risk. So this idea of the widowhood effect, right? Greater risk of morbidity and mortality in the first 6 months, 3 months, year of after the death of a spouse. So this study is really the first to do a comprehensive longitudinal examination of this first year at this scale with both capturing the psychological and biological side of potential risk. So the data I'll show you today is drawn from visit 2, which was about 4 months after the death of their spouse. And at this visit, importantly, we did a Trier social stress test with our participants. So again, most important probably context is that this is about 4 months post-loss. And so they came in early in the morning. We had a catheter insertion so that we could do multiple blood draws, but only stick them twice. They went through a Trier social stress test, which I am assuming we are all quite familiar with. It's about a 15-minute standardized stressor, asking anyone to count backwards from 2,100 and whatever it is by 17 stresses people out. They're on camera, all of that good stuff to elicit a stress response quite reliably. Then we debrief them and let them know, of course, the whole point was that was difficult, and it being difficult is not a you thing, it's a design of the study thing, and then they answer the questionnaires. So again, what we're looking at here, what I'm really interested in is how high grief symptoms might be affecting these trajectories from this baseline catheter insertion to 2 hours after the stressor. And we found that grief symptoms did promote inflammation in this sample. So the folks in the sample of 111 bereaved spouses who had higher grief symptoms had a 19% larger increase per hour in serum interleukin 6, which is causally related to cardiovascular disease progression, compared to the bereaved spouses who had low grief symptoms. And this was consistent across unadjusted and adjusted models, even after controlling for depressive symptoms. So this is really capturing the unique piece of grief symptoms that are things like yearning for your loved one. And I want to emphasize again that interleukin 6 is really involved in all of, or not all, but many of these age-related diseases that we care about. So this is the foundation that I was really excited to test, but then left me just wanting so much more, because, one stressor is not how our days go. We often face multiple stressors throughout our days. So here what I've done is just take an approximation of that trajectory that I showed you on the previous slide. So here we're pulling from the high grief group and putting that here, and then this is the low grief group. So we might imagine that over the course of a day, if you face multiple stressors, that it might look something more like this, where we see a lot more vulnerability from the folks in this high grief group through the buildup of inflammation throughout in the face of these stressors. And so that really leads me to where I'm excited to focus more on, which is thinking about these stress processes in daily life, and specifically in the context of a more recent social loss experience. And initially, I plan to focus on spousal bereavement. And the reason for that is really based on a lot of foundational work, thinking about what are our social relationships doing for us? What is lost when we experience the loss of a spouse, the loss of a loved one? And so Myron Hofer did a lot of really elegant work looking at rat pups and mothers, and identified that you could really only see how the mother was impacting and regulating that rat pup when you removed the mother from the situation, right? So he deemed these hidden regulators because they're only hidden when they are hidden until they are removed. So I love this quote from Dave Sabara and Cindy Hazan's 2008 paper. I come back to it a lot. This idea that only when attachments are disrupted can the powerful homeostasis maintaining functions of the bond be observed. So in my view, I'm excited to be thinking about more of the immediate time after or sooner in that process than a lot of the bereavement studies that are, taking folks who have been bereaved for somewhere between 6 months to 12 years and then trying to aggregate and look at things within those big samples. I think we really need to zoom in on what is lost when we lose a partner and what can that tell us about attachment, about the purpose of our close relationships in through a stress process lens, especially. So in conclusion, I want to say, please take advantage of the Stress Measurement Network resources. Reach out to me with ideas or thoughts on what would be the best resource for folks who are currently or up and coming in stress science. And I think that now is the time for all of us to really be thinking about how we can contribute to aging research and consider how stress processes might be contributing across these biomarkers of aging. And then I'm really excited about thinking about stress response processes through these times of social loss, because it offers this lens to, again, better understand how our close partners are affecting our health and potentially our aging trajectories following this loss. So I'm in the process of thinking about all of these questions, and a lot of the folks on this call I've seen and really admire. So if any pieces of these are compelling to you or you have different perspectives on them, I really welcome any contact, any criticism, collaboration, any of that. I'm happy to discuss. So I really just wanted to take a moment to say thank you to collaborators, institutions, and the organizers for setting this up. It's really special to be in a place, in an environment with so many social personality and health folks. I think there's a lot of great work that we can all do together. Thank you.
Awesome. Thank you, Ryan. That was also really interesting. We're going to open it up broadly to everybody so the speakers can discuss questions. You can feel free to drop them in the chat or raise your hand, unmute, and ask them. This is really supposed to be like your time to discuss stress and what we just heard. Okay, so awkward silence. So we have some questions that would also help. So for both of you, and you both sort of talked about this in your talks, but I'm just going to ask it anyway. In your assessment of the literature or the data that you've worked on with yourself, do you feel like we're missing anything in how we think about stress and how we're measuring stress? Again, you both really touched on this during your talk, but is there anything you want to add or talk about?
I think I guess I can go first since I'm speaking. Let me pause for a second. Ryan, do you have anything to say? No. So I think one of the things that's been really big in my conversations with collaborators is this integration piece. So thinking for example, Ryan's work and the work that she's planning on doing here soon is, integrating these daily linkages with more long-term or more potent, for lack of a better word, life experiences that are negative and stressful. I think there's been a little bit of work in that area already, but I think it needs to be more, and we need to better characterize it for what it is.
I completely agree. And I think that in the bereavement and like social loss space, I think that, I was speaking to the fact that there are so few studies that really capture this first year where we know that the health risk is the most intense, as well as the psychological experience of adjusting. So I think that capturing that first year post social loss and capturing daily processes through that is really critical. But to me, the biggest gap that we have is how high SES our samples are, especially in bereavement science, especially considering that the loss of a partner may be the loss of that income and the added cost and all of the resources that may be lost as well. So I really want to be thinking about the intersection of socioeconomic status and stress response processes in this time after the death of a spouse or divorce or whatever the sort of social loss experience is. And I do think that that is one of the biggest disservices that we have in bereavement science right now, because these are certainly the folks who would benefit the most from additional resources that are addressing stress, right? So thinking about whether that be SES at the individual level or neighborhood resources and sort of the community environment that one is living in while grappling with and reorganizing your life. There's some really good work around the age of the neighborhood that you're living in. And if the folks around you, if widowhood is a more normative experience, right? And there are just people immediately down the street that you can connect with. So anyway, I think SES, thinking about the built and neighborhood environment, and then also capturing that on a quicker time scale after the major stressor and change in relationship.
I actually have a question for Ryan if nobody else wants to go. So Ryan, you talk about needing to have a quicker, like, kind of response to bereaved families and whatnot. Do you have any suggestions for someone who's interested in maybe starting or looking into, bereaved relationships? Like, what is a good way to start potentially thinking about it to maybe get that faster
Yeah, I think that we've focused on grief a lot, which makes really good sense, but it makes me wonder what we're sort of missing in terms of other components of that experience, like health behaviors. So I think that there's a lot of space for folks with different perspectives, interests to contribute to bereavement science, especially when we start thinking about the daily level. I think that it is really critical to think. I think that it's really critical also to be with these populations very conscious of how much time and demand you're requiring of folks and the compensation. So that's something that I really want to prioritize moving forward is given the sensitivity of this time period, we really need to be paying our participants quite well, right? And so I think that what I grapple with the most is what we need to do more work in that first year, but how close is as close as we want to go before it feels ethically, morally more ambiguous. I think that I get excited about the idea of thinking more about mixed methods work that can kind of use qualitative and focus group led work to inform the quantitative questions we end up asking. So that would be something that I would really encourage folks to go towards is before we start diving in or like applying whatever we study in one population to this population. I think we would benefit from taking a more mixed methods approach in some of this work and going to folks who are going through it and really asking what are the pieces that you are identifying as really critical to your experience. And thanks, Dakota.
So we have one hand raised and one question in the chat. Let's go with Shantine first, if you want to unmute.
Yes, I'll unmute. Oh, sorry, my camera is not set up correctly. Yes, so I thank you so much for the talk, and I have a question for the first talk. I'm just wondering. Yeah, I'm very intrigued by the reactivity, recovery, pile up, and also the residual. This concept in terms of affective reaction, but statistically, how do we measure them? And how do you measure that? It's maybe like in different fields, they have different measures, but I'm curious, like how do you operationalize or capture those terms?
Yeah, that is a great question and one that is debatable depending on who you ask. So bear with me, but in my opinion, the most common way that we statistically characterize these affective reactivity and affective residue so I'll start with affective reactivity because it's easier. It's the simple covariation between stressor exposure and affect. So it's just the slope between these two things. If you're interested in understanding how, for example, reactivity informs a health outcome, most often we see people running like a multi-level model where the exposure predicts affect in some way, either negative or positive affect, and then outputting that and then using those kind of random slopes to inform the next model. Residue is very similar. The main difference being that it's the lagged association. So does a stressor in the previous day inform your affect? And it's pretty similar in the model. The main thing to remember is to include your same day affect as well, so that you're accounting for that in the model as well.
Thank you so much. That's very clear.
Does anyone want to take a stab at the chat question? It's thinking about loneliness and how long.
So I'll read it out loud. So Karina at Penn State says, an interesting thing to think about is how long a period of time of stress, loneliness, etc., is considered normative, and when it starts to become suboptimal in terms of mental and physical health outcomes, acute versus chronic experiences, and how that itself relates to aging. And I think that, and I don't know if Ryan will agree with me, I think that's something that everybody in the stress literature struggles with, is trying to understand, when does these sorts of things become like a daily experience that, it happens and then you move on, versus something that happens repeatedly and becomes chronic. So at what point does it become a chronic situation? And I don't think daily stress has an answer. That literature, I don't think they have an answer at all. So maybe the bereavement, loneliness side, social isolation piece does, but I can say nearly for certain that the daily stress side of things do not have that nailed down.
I certainly have no clear answer either, but I wanted to say that I think in thinking about bereavement or social loss experiences generally, one thing that we're looking at is like the attachment hierarchy and how you're shifting your sort of social, who do you go to when you're distressed, right, after the death of a spouse. And so in that context, it matters probably. Again, this is based on the theoretical work that we have, me hypothesizing, it matters probably how many people you already have that you can go to and trust, right? Because what we're starting to loosely see is that, and tracks with probably all of our intuition, is that probably adding new people in that first year is quite stressful. You can imagine that that's either you're going to social situations where you went with your partner and now navigating that without your partner. That probably induces a lot of negative emotions, even if you're having the positive ones too, or making fully new friends, which is hard at any point, and is itself a stressful experience. So when thinking about loneliness and bereavement, I really would love, I mean, I would love for anyone who is interested in these questions to be thinking about the social context that the loss happened in and how isolation, because, you know, maybe your partner was the main person you had social interactions with on a daily basis, right? How that difference of social environment and network may really be part of the key for understanding that loneliness piece. The loneliness, as well as on the flip side, what I think of as having positive social experiences and how incredibly powerful it is when you're in the absolute pits of your day-to-day life to have that moment of just a little spark of joy because you got to talk to someone, right? And someone who understands the situation or what you're going through in any way, or was just kind. So I really like the idea of thinking about these positive emotion experiences. And I think the flip side of that is the loneliness, isolation. So where I'm at with it is it's got to be hard to add people to your social network post loss. So how do you navigate if you are someone who is more isolated after that loss? So I just have more questions for you, Karina, but I know you'll have an amazing career answering them. So.
So I'll ask one that's not off script. So I did not give it to you before. But if you have advice for someone who's just getting into this research, what would it be, especially for junior people, for each of you? What do you think?
I have an initial kind of joke one pertaining to daily stress, and it's just Google David Almeida. Because if you just Google scholar him, all the papers pretty much pertaining to daily stress will be there. So that's kind of a joke. But it leads into a more serious answer, which is just, you know, look up people that you may know or may be interested in, who could be doing this work, and just see what they're doing. See, you know, the statistical methods that they're using, the models that they're using, the designs that they're using, and start there. Or ask one of us.
And I'll hop in there with I love YouTube videos, and there are some good YouTube videos out there that really got me. That was the only way I was learning, or that was a really key way of how I was learning as a grad student. And so that kind of motivated me down science communication routes. And so I think that what we're trying to do with like the Stress Puzzle podcast is largely have like an entry point for people who are just getting into the field, feeling overwhelmed, feeling like, you know, bringing up ideas that are 20 years old, but you don't know are 20 years old, right? So I think that I really value the idea of having this entry point that people can get into. And then the other thing I would point bereavement related folks to is a recent paper by Mary Frances O'Connor and her team, really focusing on the grieving as learning. So thinking about grief not at one time point, which is something I think we don't do often enough. So thinking about the adaptive dynamic process of grieving and what that sort of means. I think that you could generate a whole program of research just from thinking about what the dynamic process of grieving and updating your expectations about your partner's availability might be. So that's a really nice entry point to start thinking about these kinds of questions.
I think we're pretty much out of time, but thank you so much. This was really, really helpful. I really enjoyed this. I hope everyone else did too. If people want to like clap, I will like actually clap. Yay. Yeah, thanks so much. Also, the stress resources that you gave, I already sent them to my students. Really appreciate it.
Awesome. Thanks.